Hi guys, I'm glad to be taking you through this interesting course, which I'll be learning about the gene. That unit that determines inheritance and how it is expressed. So let's get started. Okay, so what makes you different from your siblings and even your friends? It's basically the gene. It's the kind of genes that you inherited from your father or your mother, uh, and in combination with your environment. But basically, this topic is about the gene. So, after this lesson, you should be able to describe the structure of a gene, should also be able to describe the process of genetic transcription and translation. So what is this gene? A gene is a nucleotide sequence in a DNA molecule that acts as a functional unit for the production of a protein, a structural RNA, or a catalytic or regulatory RNA molecule. So in essence, what we're saying there is that a gene is a nucleotide sequence in the DNA molecule that can produce two things. It can produce either a protein or an RNA molecule. And it is a DNA molecule. So here I'm showing the structure or the image of a chromosome, and then breaking it down into the DNA, and then breaking it down into the components of the DNA itself, the base pairs. So a gene is a sequence of nucleotides in the DNA, a string of nucleotides, and we will see what the structure of the DNA looks like. So the human genome, which is the totality of the genetic constitution of the human, is made up of just over 3 billion nucleotides long and divided into 23 pair of chromosomes, which is 46 chromosomes. So which means that we are saying that the human has 46 chromosomes, 22 autosomes, and two sex chromosomes. So these nucleotides are divided between those 23 pairs of chromosomes. And then the Human Genome Project, which made this available, actually has helped a lot because it has allowed us to be able to estimate the amount of genes that are present in the human genome. And it's estimated that we have about 25,000 genes in the human genome, broken down to about 21,000 protein coding genes and about 5,000 non-protein coding, which is the RNA, different types of RNA, mRNA, tRNA, etc. So what is this DNA structure? Because you need to understand the DNA structure in order to be able to understand how the gene transcription and gene translation happens. So the DNA structure is a polynucleotide of nucleic acids, which is composed of the 5-carbon sugar, the deoxyribose, a nitrogenous base, which is either can be a purine or a pyrimidine base, and then a phosphate group. Now, these two chains that run parallel to each other, are bonded together by hydrogen bonds. So you have two hydrogen bonds between adenine and thymine nitrogenous bases, and then you have two hydrogen bonds between cytosine and guanine nitrogenous bases. And so this structure allows the chemical information that is present in this DNA to be able to be transcribed into the required information that we want, the required product, which is the protein or the RNA. And so that's the basis of transcription. So from this image, you would see that this is a double helix, two chains running and parallel to each other, and then made up of the deoxyribose, made up of the phosphate group and the nitrogenous bases. And you can see the pairings between those nitrogenous bases and the hydrogen bonds in between. So basically, we have something in molecular biology that we call the central dogma. So it's just saying that, look, information is transmitted from the DNA into mRNA and into protein. So the process whereby the information in DNA is copied into mRNA, messenger RNA, is transcription. And the process by which that mRNA is copied into a protein is what you call translation. So the structure of this DNA and the sequence of nucleotide that it contains actually specifies the amino acid sequence of the polypeptide chains of proteins. this is basically what genetics taught. They look at how this sequence that they find in genes, how are these things expressed into proteins, and at that level they look at what happens at doing transcription and what happens doing